All right. Hi, everyone, and welcome back to a sort of next video on transport. I want to remind you that these videos are going to be relatively fast-paced. So if you need to ever sort of pause and take notes, feel free to pause. If you need to rewind and rewatch something, go ahead and rewind and rewatch. And remember that we're going to cover these in class, but by looking at this ahead of time, you're sort of helping yourself be prepared for the day. And so today we're going to look at transport, which is going to be the theme for our week. And so in this video, we're going to remind ourselves of the difference between active and passive transport. We're going to introduce a new idea of secondary active transport, which we will revisit during the week, but will be important for our upcoming exam. And then we'll remind ourselves of the movement of water across a membrane in osmosis, and kind of reintroduce this idea of tonicity, which will be present in our lab activity this week. So without further ado, let's jump right into it. So what we're going to do real quick is just remind ourselves of the difference between passive and active transport, and kind of go through these couple quick examples of some differences we can expect to see. And so we have passive transport and we have active transport. And as you may have already been able to guess, a major difference between the two is the lack of requirement of energy for passive transport and the requirement of energy in active transport. Now I want to be clear that the primary mode of energy is ATP, but we can also use kinetic energy. For example, we can use electrons as a form of energy. And so ATP is not the only source of energy in active transport. When it comes to passive transport, the reason why there is no energy needed is because the movement of molecules are moving with the flow of the sort of laws of physics. And so we're moving molecules from high concentration to low concentration, and that requires no energy investment whatsoever. Active transport, on the other hand, is moving molecules from low to high concentration. And anytime you're going against nature, we're going to need some energy to make that possible. And again, that doesn't have to always be ATP. There can be other forms of energy that initiate this movement. So let's take a look at our examples of passive transport, which is what we're going to get into a little bit more than the active stuff. And so what I'm going to go ahead and do is label these 1, 2, and 3 just for your reference. And I want to remind you that the main characteristic of the phospholipid membrane is that it is nonpolar. And this is important because nonpolar things typically have an ability to cross the membrane that polar or charged things do not have. And so molecules that are small and nonpolar will routinely diffuse across the membrane in accordance with their concentration gradient from high to low, and just squeeze through the spaces between the phospholipid bilayer to sort of move as they move. Now, one challenge with this is that we have no control over the movement of these small nonpolar molecules because there's no way to prevent them from sort of squeezing through the membrane. So these little purple things might be an oxygen, which is a small nonpolar gas that routinely diffuses across the membrane. Everything else, everything that is in this polar or charged category, is going to require the facilitation of a protein in order to get across the membrane. And so regardless of what these individual squares and circles are, we know that they have properties that require facilitation. And that's why we call it facilitated diffusion, not because energy is required, but because we need a protein to get these across. Now, these proteins come in two flavors. There are channel proteins that are just open channels that allow the same kind of diffusion qualities of simple diffusion across the membrane. It's just through a channel instead of across the membrane. In example 3, we have a gated channel that is currently closed and preventing these molecules from coming across. When this channel opens, then diffusion can continue, again from high to low concentration. So is energy needed to make this happen? No. The other thing I just want to recognize is that passive transport will continue until the system reaches equilibrium, and it's where we have equal concentration of molecules on both sides. In this example of active transport on the left, we'll give it example 4 here. We see the inclusion of ATP in this process, which tells us that we're moving from molecules from low concentration to high concentration. And in the case of these square-shaped molecules, that is precisely the case. We are seeing a higher concentration inside the cell in the direction of movement, and so by definition, energy will be required. All right. So from here, we're going to talk through a quick example of passive and active transport being utilized together under this umbrella of secondary active transport. And so what I want us to focus our attention on in the beginning here is the bottom half of this drawing, and specifically this proton pump, which is moving hydrogen ions from low concentration inside the cell to high concentration outside the cell. Because we're moving from low to high concentration, we absolutely need the inclusion of energy, and in this case we are using ATP energy. Now, this is active transport with a purpose, meaning that because we've moved these hydrogens to the outside of the cell, they have a larger purpose and they're going to do something. And in the context of this particular example, these hydrogens are extremely uncomfortable, and they're going to want to establish equilibrium. They're going to want to get back to their initial state. And so our cells will take advantage of this high concentration and allow the hydrogen ions, the protons, to diffuse back across the membrane through facilitated diffusion. Except now the energy stored in their kinetic movement is going to be utilized for another goal in mind. And in this case, it's going to be for the co-transport of sucrose from low concentration to high concentration to maximize the absorption of sugar in the cell. And so in this particular example, we have a utilization of energy in this at the primary source to move hydrogens across the membrane. But then the energy of the movement back across through the diffusion of hydrogen actually allows for this protein to capture a little bit more glucose or a little bit more sucrose across the same protein and end up with a higher concentration of sucrose inside the cell. And this is a very common theme. This can be done through the sodium-potassium pump, for example, as well as other H plus proton gradients. But the creation of a small ion gradient is often a very useful source of efficient use of ATP to help get these larger molecules across against their gradient and maximize their absorption. And so we'll see other examples of this in class this week and talk through them. But I wanted you to see how these two different kinds of transport can work together. Finally, what I want to look at here is the movement of water, the osmosis, which is the diffusion of water. And I want to talk about how we know the direction that water is going to move. And this is because water has a set of balanced concentrations. It's the concentration of solute over the concentration of solvent. And when this ratio is out of balance, it allows us to say whether or not water or solute are in higher concentration. So in this particular example, if we look at the left-hand side, the number 1 side of this diagram, we'll see all these blue dots indicates that we have a high water concentration and a relatively low concentration of solute, the red dots. On the right-hand side, we have a relatively low concentration of water and a relatively higher concentration of solute. So the question is, what direction is water going to flow? And water typically flows from high water concentration to low water concentration. So water is going to flow in this direction. Sometimes we have this sort of misunderstanding and say that water is traveling from low to high, but really what we're saying is that water is traveling in the direction of low solute concentration to high solute concentration, not along its own concentration gradient. So this is passive transport because water is traveling from high to low water concentration. Salt, if it had the opportunity, would travel in the opposite direction until we establish that sought after equilibrium. So why does this matter? When it comes to cells, the environment sort of dictates the movement of water in and out of the cells. And you may remember these terms: hypertonic, isotonic, and hypotonic. And each of these terms refer to the environment that a cell finds itself in. Hyper meaning more, iso meaning equal, and hypo meaning less. And so what this is saying is that the solution surrounding the cells is more tonic, it has more salt, higher osmolarity than the cell, whereas hypotonic, you have less salt in the surrounding environment. Think of walking into fresh water versus walking into salt water, for example. So what this does is this influences the movement of water in and out of the cells and can change the behavior of the cells themselves and provide different challenges for organisms. And so if we have a low water concentration on the outside and a high water concentration on the inside, we're going to see the movement of water leaving the cell as water chases solute. That's a big phrase we should take. In an isotonic solution where the water concentration is equal inside and outside of the cell, we don't see any change to the cell shape because the water is moving in equal proportions in and out of the cell. And then finally, when we place, when the cell is more salty than the solution, we'll see that water from the solution will travel into the cell in order to establish equilibrium, and that can actually cause cells to burst, especially if there's no cell wall to maintain rigidity. This is also important for fish, for example, that live in different saltwater or freshwater environments. If you can imagine, freshwater salmon, for example, don't really have to drink a lot of water because they are naturally saltier than the environment. We'll actually see that they absorb much of their water through their skin. And so they need to pee a lot in order to maintain water balance. But again, organisms in their environment, the goal is to maintain water balance. And so if they're in a particularly hypotonic environment, then they need extra consideration when it comes to making sure they don't have too much water in their system. When we're talking about saltwater fish, for example, they live in a hypertonic environment. So they're constantly worried about water loss. And so they have adaptations that maximize their own water balance, for example, and that challenge their osmoregulation. For example, they're worried about moving stuff out, and so they are trying to maintain their ion balance so that they can maintain their water balance. And they get rid of concentrated urine so that they're not losing too much water to their environment. So that's all we're going to do with this here. I'll see you later.